All right, Bismillah. Assalamu alaikum. So let's see how this works. Um, I'm going to start again from chapter 7 that we have already spent some time studying in our class already. Um, so again, we're talking about enzyme controls. We're talking about how enzymes can be regulated. As I said last time, there are basically there are basically uh, four approaches of doing this. One is called the allosteric control. Second one is called reversible covalent modifications. Third one is called proteolytic activation, and the fourth one is uh, multiple forms of the same enzyme. And this is basically done at the level of DNA. So certain different forms of the genes are expressed. So you may have slightly different versions of a gene being made in different tissues. We will not talk about this in our class, and we'll basically focus on these three major forms of enzyme regulation. And we've talked about allosteric quite a bit. Um, we'll talk about reversible covalent modification as well as proteolytic activation going forward. So again, we said allosteric enzymes are enzymes whose activity can be affected, can be controlled by binding of molecules, and we call them effectors. And these molecules can be substrates or inhibitors or activators, and they usually result in a conformational change in the quaternary structure. So again, this is a keyword to understand that allosteric enzymes are enzymes that have quaternary structures. So they not only have the primary and secondary in the tertiary structures, but they also have a quaternary structures. And a point to note here is that all allosteric enzymes show cooperativity, and we have seen this with hemoglobin before, how binding of hemoglobin is regulated by binding of other oxygen molecules, and that's called cooperativity. So uh, cooperativity describes the trends that occurs when enzymes or proteins contain multiple subunits, multiple binding sites, and it describes the chain that occurs when binding um, site of one of the structures is activated or deactivated by a result of binding of other sites. So the example that we gave last in the past in our class was the example of, for example, oxygen O2 binding to hemoglobin. When it binds to one subunit of hemoglobin, the binding affinity of oxygen for the other subunits increase, and that is a positive cooperativity. Again, the word cooperativity, it will be a positive cooperativity. Um, so it also described again in the case of hemoglobin was the binding affinity being changed. So it describes increase or decrease of affinity for binding, right? So again, we talk about allosteric enzymes. We're saying allosteric enzymes are enzymes um, in which effector molecules change activity or the affinity of enzyme by binding at a second site, and that's where the key word for allosteric. When we talk about allosteric enzymes, uh, we're talking about something binding to a different site. So here's an example where you have an active site shown here, and the active site starts off distorted. It's not proper. The substrate cannot bind to it. There's no binding happening here. Um, and but when something binds to a different site, which is the allosteric site, so you have an effector molecule, in this case it's an activator. When this guy binds to it, there is a conformational change, you can see, and now the substrate is able to bind, and you have a functional enzyme substrate complex, um, and then which will give you enzyme plus product, right? Um, so this is how things will work. Now also you can have the same slightly different, so this is called activation where things are activated. You can also have inhibition where things are inhibited, so where the enzyme actually has a functional binding site and it's active, it could work, but in the allosteric site you have something a modulator binding, which is in this case an inhibitor, and controlling inhibiting the enzyme activity. So you can have effector molecules speeding up enzyme activity, and we call them positive allosterism, or you can have molecules slowing down the enzyme. In this case, we call them negative allosterism. Okay, so again, allosteric sites allosteric enzymes are enzymes that have allosteric sites. Um, and again, they have to be oligomers. They have to have a quaternary structure, right? Um, and then binding of something changes the conformation of the quaternary structure, and they can be inhibited, they can be activated. Now, a lot of it is a lot of this notion of allosterism or allosteric regulation has been studied using this enzyme called ATCase. And so ATCase is an enzyme that is involved in a pathway, metabolic pathway, and this enzyme is an allosteric enzyme, and this enzyme is used in what is called feedback inhibition of that pathway. So the pathway is controlled by this mechanism called feedback inhibition, in which ATCase is used as a main enzyme, which is regulated by allosteric regulation. And we talked about feedback inhibition a lot, so please take a look at this link, which shows this video here, and you'll see what this point of feedback inhibition is. Okay, now I talk about allosteric regulation. The effectors, the compound that bind to it, they could be homotropic modulator or they could be heterotropic modulator. So let's start with heterotropic first. In heterotropic modulator, these are compounds um, uh, or they're regulatory molecules that are not the same as the enzyme substrate. Say they are different compounds. So for example, hemoglobin, remember hemoglobin, what does it bind mainly? It binds oxygen. So we say oxygen is a main substrate for it. But you can have compounds like H plus or CO2 or this thing we talked about 2,3-bisphosphoglycerate. So these guys can bind to hemoglobin as well, and binding of all of them to hemoglobin will affect somehow its ability to bind to oxygen. And since these guys are not the same as its substrate, we call them heterotropic. Hetero means different, heterotropic, right? In some cases, we have things that are called homotropic. So homotropic are effector molecules, binding of which affects the binding of the same molecule to other subunits. And here the example really is, if you look at hemoglobin tetramer, now all of them bind oxygen molecules. So we know the binding of one oxygen molecule to one subunit has an effect on the other subunits. So in this case, oxygen is both a regulator as well as a substrate. So oxygen in this case is considered to be a homotropic regulator because it is a substrate for the target enzyme, right? So oxygen is the case. Okay, so please make sure you understand the difference between the two. Most of the time we talk about heterotropic because that is the vast majority of allosteric regulators, but in some cases homotropic regulators are also important, like oxygen binding to hemoglobin. Now, we have seen enzyme kinetics in preceding chapters, and we know that if you draw this V naught versus substrate concentration graphs, we see this Michaelis-Menten graph, and it becomes like this, and from there you can get the Vmax and you can get the Km and so on and so forth, right? And this is called a hyperbolic curve, right? Now this is for non-allosteric enzyme, so non-allosteric enzymes. This is how they normally work. But if you have an enzyme that is allosteric and that shows cooperativity, right, or allosterism, then you find they have the sigmoidal curve that looks like an S, right? And this is called the sigmoidal curve. Okay. Um, and so you can see the difference between a normal non-allosteric enzyme or normal enzyme. We call them Michaelis-Menten. And if you have an allosteric enzyme, we call them that they have sigmoidal curves. And that's the same thing shown here, that enzymes that follow Michaelis-Menten, they show this hyperbolic curve, right, which you've seen before, and allosteric enzymes show a sigmoidal curve, which shows allosterism. So again, anytime you have a sigmoidal curve, you're showing allosterism or cooperativity, allosterism as well as cooperativity. Sorry, my writing is very big. Okay, cooperativity. Okay. So again, cooperativity is, again, we talk about this a lot. Binding of one molecule increases affinity for other molecules, and we have seen this with hemoglobin and oxygen a lot. It could be positive if the affinities increase, or if binding of one molecule decreases the affinity, it's called negative cooperativity. And so, for example, this is a substrate, this is an enzyme, so it binds to it. Now, if binding of this enzyme, substrate to the enzyme, if it increases the activity of other guys to bind, this would be an example of positive cooperativity. If binding of the substrate to the enzyme decreases the affinity for this guy to bind here, and this would be an example of negative cooperativity. Right? So I hope this makes sense. All right. Um, Again, positive, substrate binding to one site stabilizes the conformation, and then it makes it, bind to the other ones more. Some forms can be negative as well. Okay. Now, if you see how the graph would look, right? How a typical uh, allosteric enzyme would look. It would look like. Oops, sorry, delete that. So how it would look basically like the normal graph. It would look sigmoidal graph like this. Okay. Now, if you have a positive allosterism, if you have an activator going on, the activator is going to change the graph, so you'll have something like this. The graph is going to move to the left hand side, and you'll find the graph looks like this, right? If you have a negative allosterism, it's going to look, it's actually different, and the graph is going to shift to the right, right? And that's shown here. It's going to the right. So make sure you understand how the positive and the negative allosterism would be reflected in a sigmoidal graph example. Okay. Now this is bringing us back to this notion of this enzyme ATCase, and we talked about this earlier, that this is an enzyme which is regulated by feedback inhibition, and it's an allosteric enzyme. So if you saw the video before, feedback inhibition is if you have a pathway that is multi-step. So you have, for example, you have glutamine, for example. Glutamine becomes carbamyl phosphate, which becomes carbamyl aspartate, which becomes dihydroorate, UMP, UDP, UTP, and CTP. And CTP is the final product in the pathway. So in feedback inhibition, the final product of the pathway, this compound, which is again a product, it served as an inhibitor of an enzyme earlier in the pathway, right? It stops an enzyme which is earlier. It does not stop immediate enzyme. That does not happen. This is not feedback inhibition. Feedback inhibition is that you have to have an enzyme which is earlier in the pathway which is affected. And the whole idea behind this is, if you have, so let me delete all this. Suppose you have, you know, um, you have too much of this product form. You have too much of CTP, right? And it's wasteful for the cell to keep making CTP. You don't want it to keep making CTP. So what does the cell do? So if you have too much CTP, right? If you don't need it, well, stop it. Well, you can stop here. If you stop here, then by inhibiting this enzyme here, then you have a lot of CTP. So if you stop this enzyme, then you have a lot of CTP being left, right? And that's not good. If you stop here, for example, then you have a lot of UDP being formed. If you stop here, you have a lot of UMP. If you stop here, and so on and so forth. So why don't you stop the enzyme which is unique to that pathway? Meaning this is not needed, this is not needed, right? This is not needed. So none of these things are needed. So stop the first enzyme that stops things that are not needed, right? And that is why it's called feedback inhibition, where CTP would go and bind to ATCase, but it'll bind to a different site. It's going to bind to an allosteric site. So it's going to bind to an allosteric, so allosteric site, right? And of course there will be an active site here. This will be active site, which will be different. and that's the active site, and that is really the main difference between main point of allosteric regulation is that something binds to allosteric site. So CTP would be the allosteric regulator and it'll bind to the allosteric site. So based on this, um, we can see there's a negative sign here. If you look closely, there's a negative sign here. So CTP is a feedback inhibitor. In this case, we see a positive sign here. So ATP in this case is a positive allosteric activator. So this enzyme ATCase is activated by ATP as inhibited by CTP. And both ATP and CTP, both of them are binding to the allosteric site, not to the active site. And if you look at this pathway here, uh, I would say the active site binds carbamyl phosphate. That's the active site. This guy binds to the active site, right? And ATP and CTP both are binding to the allosteric site. That's shown here. Okay, I hope this makes sense. And since ATP is an activator, we said the activator is going to show a right shifted graph, sigmoidal graph. Sorry, a left shifted sigmoidal graph. Shown here in red now, and since CTP is an inhibitor, it's going to show a right shifted sigmoidal graph, and that's what you really see in real life, that ATP so control ATCase is sigmoidal. It shows normal behavior like that, sigmoidal graph, and since CTP is a negative inhibitor, it shifts the graph to the right. ATP is an activator, shift to the left hand side. Okay, and this is just to show you, don't have to memorize this. Oh, by the way, you don't have to memorize any of the other pathway and pictures we talked about. Okay, so this is the example of this is just showing you the structure of ATCase. ATCase is a oligomer, right? And we talked about this, that is an oligomer. It has many subunits, and we said oligomers. We said any enzyme which is regulated by allosteric regulation has to be an oligomer. So you find um, it has 12 plus 18, so it's pretty big. It's got 6 subunits. Um, so you have 3 uh, dimers. So you have dimer and dimer and dimer. So these are all regulatory subunits. So you have 6 of those guys, and the catalytic subunits are uh, you have trimers. So you have 3 linked together, and there are this is one trimer, and then there's underneath one underneath. So you have 6 of those. So you have 6 plus 6. You have 12 subunits total, and that is the quaternary structure for this. And that, again, there's nothing to memorize. Just showing you that ATCase is an enzyme which is regulated by allosteric regulation, and it has oligomeric structure.